Hi, I'm a zombie. Why? Because today I'm going to be talking about DNA. And with DNA comes mutations.
Today we're going to be talking about protein synthesis and how we start out with DNA and end up with a protein. First, we need to talk about what DNA and RNA is. DNA has a sugar phosphate backbone just like RNA, but it is long. Unlike RNA, RNA is short. DNA is also double-stranded, while RNA is single-stranded. They both have 4 nitrogenous bases, but the main difference is that RNA has a different 4th nitrogenous base. Since DNA has the sugar deoxyribose, it has thymine, but since thymine cannot bond with the sugar molecule of ribose, RNA must have uracil. Assuming you already know about DNA, then you should know that the nitrogenous bases pair up with each other. Adenine with thymine, guanine with cytosine, except for with RNA, instead of thymine, there's uracil. Since you know this, then you should know that they create a certain code. The code on the DNA molecule is copied by an mRNA molecule during the process of transcription. Before the mRNA molecule can leave the nucleus, it must go through a process called mRNA processing. There's 4 parts of the mRNA molecule. There's exons, introns, a cap, and a poly A tail. Exons are what we want to keep. It's the codes that we use to make proteins. Introns, we don't need that, so we cut that out through our mRNA processing. There's also a cap at the beginning of the RNA strand and a poly A tail at the end. Those codons we're talking about are 3 codes, so a guanine, an adenine, and a cytosine would make up 1 codon. We have what we call start codons and stop codons. A start codon tells us where it starts, and a stop codon, well, it tells you where it stops. Another type of RNA we have is called tRNA, or transfer RNA. It has anticodons, which are what it sounds like, the opposite of what the other codons are, and also brings amino acids to the RNA. rRNA, or more likely known as ribosomes, is where all of the RNA is made up at once. An rRNA has 2 subunits. The most important thing about rRNA is the mRNA binding site. That is where proteins are made. mRNA is read there, and the tRNA comes and brings the amino acids in a certain code that makes up a protein. The process of which proteins are made is called translation, which this picture depicts it better than I could explain it. Here's my favorite part. This is where we get to talk about the mutations. First, there's a point mutation. That is where one nucleotide is different. A silence mutation is one that doesn't affect the protein. The codon changes, but it's the same amino acid. Like I said, there's codons, and certain codons code for a certain amino acid. Well, multiple codes code for one amino acid. So if one changes, but it still means the same amino acid, it doesn't make a difference. Another type of point mutation is missense mutation. The nucleotide is different, the wrong amino acid is created, thus creating a different protein. Nonsense mutation is where the nucleotide changes and it changes to a stop codon, thus stopping the protein prematurely. The worst kind of mutation is called a frame shift mutation. That's where a mutation where a nucleotide shifts the whole entire protein. There's either an insertion or a deletion. An insertion is where a nucleotide is inserted somewhere, thus throwing off the whole entire pattern, or deletion is where a nucleotide is taken out for some reason. This could cause a body not to have insulin. It can also cause a body not to have lactate. It can cause a lot of different things, not just, as most people think, mutations as 3rd arms and 16 eyes or something like that. That is mutation. Not all mutations are as bad as they seem. Some mutations could possibly even be good. Did you know blue eyes is a mutation?